Today I am going to discuss Vacutainer tubes. Hi everyone, I am Barman. Vacutainer tubes are also known as blood collection tubes. There are different types of Vacutainer tubes used in the laboratory for various diagnostic tests. Each tube has different color and different functions. You have to select specific tube for a specific test. Vacutainer tubes are made up of glass or maybe made up of plastic. Inside the vacutainer there is a partial vacuum by a rubber stopper. The air pressure within the vacutainer is negative pressure which compared to the environment outside. After inserting the needle into the vein, the phlebotomist pushes the tube into the holder. Let's zoom it here. The other side of the needle is penetrating the rubber stopper of the tube. And there is a negative pressure. Because of that negative pressure, the blood within the vein will get into the tube. Because the pressure within the vein is more than the tube. And the tube is having negative pressure. Because of that, the blood will get into the tube. And the vacuum is in such a way that only required volume of blood will enter into the tube. If you need 2 ml of blood for a particular test, then only 2 ml will be collected. If you need 1.6 ml of blood, then 1.6 ml of blood will be collected into the vacutainer. Different types of blood test requires different types of blood specimen. Most of the tube has additives, which are also called as anticoagulants. Anticoagulants are chemicals which prevents blood clotting. Anticoagulant. Anti means against or opposite. Coagulant means clotting. If we combine both, it will be preventing the blood clotting. Anticoagulant, which prevents blood clotting. There are 13 blood coagulation factors. Let me briefly explain the clotting process very briefly so that you can understand. When clotting will start, it will activate one of the 13 coagulation factor. It will get activated. Then once it is activated, that factor will activate the other factor. Again another factor will get activated. This factor will again activate other coagulation factor like this. It will go on. It will activate other coagulation factor. This is kind of chemical reaction which one activate another, and eventually there will be fibrin clot. If you will get the fibrin clot, the clotting process is complete. The blood will become solidified and it will stop the bleeding. This is the normal phenomena of clotting. But what happens when we use anticoagulant? Anticoagulant will remove one of the clotting factor and you will not be able to complete the clotting process. Let me explain how anticoagulant works. When clotting starts, it will activate one factor. The activated factor will activate the other factor. It will activate other factor. But suppose the anticoagulant act on this factor, it will make unavailable of this factor. The factor is no more in the blood. So this factor is being activated. It can activate only the next factor, not this factor. So it cannot activate this factor bypassing this one. So the fibrin clot will not form because of unavailability of one of the 13 coagulation factor. Anticoagulants acts like that. And remember, different anticoagulants will act on different clotting factor. Their function may be different. And remember, there are some tubes which doesn't contain any anticoagulants, like this red tube, which is also known as plain tube. It doesn't have any anticoagulants, and each tube has different functions, so use it according to the test recommended by the physician. First, we are going to discuss about the lavender tube. The additive in the lavender tube is ethylene diamine tetraacetate, which is known as EDTA in short. Along with EDTA, there may be potassium. It may be dipotassium, that is K2-EDTA, or it may be K3-EDTA. Remember, the ratio of anticoagulant and blood has to be proper. If insufficient anticoagulant will not prevent the clotting properly, there may be a microclot. You need 1.5 to 2 milligram of EDTA in 1 ml of blood. This is the calculation. These tubes your company made tube, and they used to keep anticoagulant for 2 ml of blood already determined. So you do not need to bother about that concentration of the anticoagulant. You just need to select the proper tube. What are the functions of EDTA? Already I have discussed that anticoagulant act either one of the clotting factor, it will unavailable the clotting factor, and it will stop the clotting process. How EDTA works? EDTA will bind with the calcium ion, and it will form calcium salt. The calcium ion is unavailable in the blood. The calcium is the fourth clotting factor. If there is unavailable of fourth clotting factor, the clotting process will not proceed further. EDTA is used to prevent clotting of whole blood for hematological investigation. For the basic hematological investigation, we use EDTA. As EDTA, it preserves the cellular component very well. So, EDTA will not alter the morphology of the blood cells. So, all the cell count, where morphology of the blood cells are important, we use EDTA. Basically, it's hematological test. What are the uses? Which test you can use these tubes? You can use these tubes in various tests like complete blood count. Complete blood count, you can use lavender tube, which contains EDTA. Already I have told you that the EDTA preserves the morphology of the cell. So in the peripheral blood film examination, you are going to observe the blood cells. So it is a perfect vacuitainer for peripheral blood film. In short, we can say it as PBF. If you are going to perform retics count, that is reticulocyte count, you need to use lavender vacuitainer. If you are going to perform HbA1c, which is also known as glycated hemoglobin. This test is done for a person with diabetes. So I have already told you that the proper proportion of anticoagulant and blood is important. Like that, you need to mix the anticoagulant and blood properly, so that there will not be any microclotting. For correct mixing of lavender tube, after collecting the blood, is invert the tube around 8 to 10 times. So it will be the proper mixing, correct mixing. Now let's move on to the grey tube. Grey tube contains 2 additives. I told you that the tube contains additives. Some tubes do not contain additive. These two contain 2 additives. One is anticoagulant and the other is not anticoagulant. It has some different functions, I'm telling you. The anticoagulant present in this tube is potassium oxalate. It prevents the clotting. It acts as anticoagulant and it will prevent the clotting process. Let's talk about the other additive. Other additive is sodium fluoride. And most importantly, the sodium fluoride, it prevents the enzyme enolase. What is the importance of enolase? Enolase is an enzyme. If you inhibit the enolase, it will eventually inhibit the glycolysis. What is the glycolysis? Breakdown of glucose. So why it is important to prevent glycolysis in this tube? Because this tube is used for estimation of blood glucose. So if you inhibit glycolysis, the red blood cells present in the specimen cannot utilize the glucose. As a result, the glucose will not break down till the test. What is the uses? Just remember G for gray and G for glucose. You can easily remember G for gray, G for glucose. We use gray tube for estimation of glucose. Other than glucose also we can use estimation of lactate and estimation of OGTT. What is OGTT? It is oral glucose tolerance test. You are mixing the tube by inverting the tube into 8 to 10 times. It is the correct mixing. Invert the tube 8 to 10 times. Next, move on to green tube. Green Vacutainer contains additive, that is heparin. There are two green tube. One is dark green and the other is light green. So both this tube contains heparin. It is a basic thing, but dark green contains sodium heparin and the light green contains lithium heparin. Remember, L for light, L for lithium. Do you know the heparin is a naturally occurring anticoagulant? Let's talk about the function of heparin. What are the functions of heparin? Heparin develops antithrombin complex. Once antithrombin complex is formed, it inhibits the formation of thrombin. The antithrombin complex also inactive the factor 10. So in this way it prevents the coagulation process. Green Vacutainers are basically used for blood chemistry. Tests like ABG. What is the full form of ABG? ABG is arterial blood gas. Arterial blood gas. And it is used for OFT. What is OFT? Osmotic fragility test. How to mix the tube after collecting the blood? Mix by inverting the tube 8 to 10 times. It is quite common. Almost most of the tubes mixing is inverting by 8 to 10 times. Next we are going to talk about the blue tube. The additive within the blue tube is sodium citrate. But what is the strength of that sodium citrate? The strength is around 3.2% sodium citrate. And the sodium citrate is a liquid anticoagulant. So what is the ratio of anticoagulant and blood? The ratio of anticoagulant and blood. That means we are going to take 1 part of anticoagulant and going to mix with 9 parts of blood. For example, the tube basically contains 0.2 ml of 3.2% trisodium citrate and 1.8 ml of blood is mixed in that tube. It is a vacuum tube. Partial vacuum is there within the tube. Once you will collect, automatically 1.8 ml of blood will be coming into the tube. And with the total volume of blood and anticoagulant, it will make up 2 ml. So in this tube, around 0.2 ml anticoagulant, then the blood is 1.8 ml. So the total is 2 ml. What is the function of sodium citrate? Sodium citrate will also bind with calcium. It will form some other complex. Eventually, the calcium is unavailable in the blood. So, the blood will not clot. Unavailable of the calcium, because of the unavailability of the calcium, it will prevent the blood clotting. Blood will not clot. Function of sodium citrate is almost similar with the ADTA. Why I say it almost similar? Let's see. Anticoagulation with ADTA is irreversible. Reversible means it can come back to its original position. Irreversible means it cannot come back to its original position. But anticoagulation with sodium citrate is reversible. That is a very important point. Once we will come to the uses, I am going to tell you this. How to mix the tube. Let's talk about the mixing. Blue Vacutainer contains 3.2% sodium citrate. From this, we can say that like invert it 3 to 4 times. 3.2% sodium citrate, 3 to 4 times inversion. So it will be easy for you to remember. Let's talk about the uses. So I told you the anticoagulation by sodium citrate is reversible. So it is used for coagulation studies. This is a very important point. For using coagulation studies, you need sodium citrate. Otherwise, it won't work. Because the anticoagulation with the sodium citrate is irreversible. Like we can use in PT. PT means prothrombin time. Then APTT. APTT means TT. TT means thrombin time. FDP. FDP means now let's talk about the black tube. Black tube is also known as ESR tube. ESR is erythrocyte sedimentation rate. Black tube also contains sodium citrate, but the strength, the percentage is different. It contains around 3.8% sodium citrate. This is a liquid anticoagulant. And what is the ratio of anticoagulant and blood? 1 part of sodium citrate is mixed with 4 parts of blood. 0.4 ml of 3.2% sodium citrate is already there within the tube, and we need to add 1.6 ml of blood, and with a final volume of 2 ml in the tube. What are the functions of sodium citrate? Previous tube was the blue tube, which also contains sodium citrate. In the black tube also contains sodium citrate. Only difference is the strength is the difference, the percentage. In black we use 3.8%, in blue we use 3.2% sodium citrate. The function of both the anticoagulants are same. Its function is to remove the calcium from the blood. It will bind with the calcium ion in the blood, and it will form some other complex, and it will make the calcium ion unavailable, which will prevent the clotting. How about mixing? Mix the tube 8 to 10 times. In case of blue, 3 to 4 times. In case of black, 8 to 10 times. Next, going to talk about the red tube and the serum separator tube, which is very important. If you need serum for your examination, then the blood has to be clot. Otherwise, we will not get the serum. In previous tube, which we have discussed, our aim was to prevent the clotting. But in the red tube and the serum separator tube, our aim is not to prevent the clotting. Here we allow the blood to clot. To get serum, the blood has to be clot. And the reason why I am discussing both the tubes together is both the tubes can provide serum. So let's talk about the differences and similarities of both the tubes. The red tube, both the tube, red tube and SST, both the tube has clot activator. Why clot activator? Our aim here is to clot the blood. So if we use clot activator, that means it will enhance the clotting process. The clotting process will be increased by using clot activator. So both the tube has clot activator to enhance the clotting process. But there is a gel present in the SST tube, that is serum separator tube. A gel is present. This is the gel. But in red tube, there is no gel. And remember, the red tube is also known as plain tube. Let's talk about the gel. What is the role of the gel in the SST tube? Let's talk about the SST tube. After collection of the blood, the gel will be in the bottom of the tube, and on top of the gel, blood is there. Wait for some time to clot. After clotting, you need to centrifuge. Okay. Let's look at the scenario after the centrifugation of this SST tube. So after centrifugation, the blood cells will be settled at the bottom. The gel will be there in between the blood cells and the serum. Because the blood cells has more density, so it is settled to the bottom, and the gel has a density between the serum and the blood cells, so it placed in between the serum and the blood cells and act as a barrier between the serum and the blood cells, so the serum won't be mixing with the blood cells. That is why this tube is known as serum separator tube, and the gel is known as serum separator gel. Let's talk about different uses of this both these tubes. Uses are same, red and SST tube. We can use this tube in serological test. All the serological tests like Widal, ASO, RF, HBS, AZ, etc. Widal is for detection of typhoid, paratyphoid. ASO is entry stapulation O. RF is rheumatoid factor. HBS is AZ hepatitis B surface antigen, which is also known as Australian antigen. In case of liver function test, you can use this plain tube and SST tube. Lipid profile test also, and serum urea, serum creatinine. And remember, most of the tests, all the tests which require serum, we can use either plain tube or SST tube. How about mixing? So many people will tell that the plain tube contain nothing, so no need to mix. But clot activator is there. You can mix by inverting the tube 5 to 6 times, so that the clot activator can get mixed with the blood and start the clotting process early. The next tube I'm going to talk is pink tube. Pink tube contains K2EDTA. The pink tube is specially used in the blood bank during the cross-matching. So during cross-matching you need two things, like one is red cell suspension, and on the other side you need serum. To get the red cell suspension, we use this pink tube, and to get the serum we can take either plain tube or SST tube. How to mix? Need to invert a tube 8 to 10 times to mix. And I told you that the serum is prepared using the plain tube. Thank you for listening till the end. Please like, share and comment. Those who are new to my channel, please subscribe to my channel. Keep supporting, it motivates me to make more such videos. Thank you.